So thank you for joining us, whatever time of day it is. I know it's quite late for us in the UK and quite very early for you guys in Australia, for Sandy and Brian. So thank you for joining us. It's trying to get everyone across the globe can be tricky sometimes. Um, we wanted to do this webinar as it's quite an exciting partnership between VSNI and MateSel. I think in the time of where things like genomic selection and environmental pressures are building. Um, MateSel offer a really exciting tool that can help you achieve your rates of genetic gain whilst also increasing that diversity that give you higher accuracies if you have a diverse population for tools for things like genomic selection, or allow you to switch your breeding programs to different focuses as new pressures arise. And then obviously from the VSNI side, being able to handle these complex data sets and give you more accurate EBVs to put into MateSel can give you a big advantage as well. So I'm going to keep my side very brief. If you do have any questions, please feel free to put them in the Q&A section or the chat below, and we will get to them as the webinar progresses. And I'm going to very quickly introduce both Sandy and Brian Kinghorn. Sandy Kinghorn brings 25 years of software and design and development experience across the education, health, and animal and plant breeding industries. He has held roles ranging from tech lead and CTO to product manager and general manager. But his real passion is working directly with end users to design intuitive and automated solutions that can make complex workflows simple. When he's not solving software problems, Sandy can be found playing soccer or football if you're in Europe, mountain biking, or on beaches of northern New South Wales, Australia. Brian Kinghorn is a quantitative geneticist with experience across a wide variety of breeding industries. Research emphasis is on design of breeding programs, including the optimal use of genomic and reproductive techniques and integration of technical, logistical, and cost issues in a single decision-making framework. Brian has a full academic background, having supervised over 41 PhD plus 25 master's students. He is an emeritus professor of quantitative genetics at the University of New England, Australia, but is also a mechanic for old cars and motorbikes, with even more YouTube hits for such endeavours than he has for breeding tools. And quickly, a bit of background on MateSel, which was originally developed by Brian in the 90s as a simple OCS tool. MateSel has evolved over the years in response to animal and plant breeding industry requirements to support additional core features such as trait/EBV targets, genetic marker targets, inbreeding targets, and managing bisexuality. Since 2020, amongst many improvements to core functionality, the following new features have been developed: committed matings, multiple end uses, mixed mating groups or open pollination, genomic mate selection, and JSON formats, inputs and outputs for integration with other systems. So with that, it's my pleasure to hand over to Sandy and Brian, and I hope you enjoy this webinar as much as I will. Thank you.
Thank you, Owen.
Thanks, Owen. Just let me know when you can see that.
Yep.
Yes, all good. Yeah, thanks, Owen. And big thanks to the wider team at VSNI for helping partnership with this webinar. So today we're going to have a look at what MateSel does, how MateSel works, and we'll go through a quick demo using the software. And we've already gone through, who we are, but I'd like to say also, obviously the true hero behind MateSel is Brian, aka Dad, I'd also like to say that the success of MateSel isn't just from the two of us and also some of the people that help through contracting. It's really through the users that really drive MateSel. We're really listening to a lot of feedback from what we get from animal and plant breeders who use the software to sort of dictate where we push the product. So really a big thanks to everyone who's already using it to help guide us in terms of where we need to take it. Okay, so what MateSel does. So as a breeder, you've got a lot of different constraints and objectives that you need to work within when you're looking to do the next round of crossings. So on the right-hand side, we've got a few examples that that typically come up within breeding programs. You might see some missing here. But you would have your EBVs that you want to push in certain directions. You want to control inbreeding. You want to make sure that genetic diversity is looked after. And you might actually have diverse product needs, especially in plant industry. You might have a species like tomatoes and then have varieties. And you're constantly looking to push different traits for different regions, for example. So there's lots to consider there. And what MateSel does is it allows you to take all of those requirements and push them through a single decision framework. So it's not a rules-based system where you step with different rules. We allow the algorithm within MateSel to take in all of those desires and let you know what your possibilities are with the genetic resources you have at hand. And it also puts your data into action. And what we mean by that is you've already gone through a lot of effort using tools such as ASReml to develop your EBVs and your selection indexes and the like. And MateSel really augments that process to take that information and show you what you can do with it essentially in terms of crossing lists. And what MateSel does, it does all of those, it achieves that single decision framework based on your desired outcomes. So you basically set your targets and MateSel will move towards those goals. So as we'll see in a second, when we look at MateSel, all of the graphical outputs that you're looking at are all to do with the progeny that will result from the crossing plan that MateSel has calculated. So what that allows you to do is really see what on screen what is going to come out, and you'll see things that you really like, maybe in terms of an EBV that's getting a really good response. But then you might see other things which aren't so good. Maybe it's the diversity or inbreeding levels aren't so good. And when you see those outcomes, you can start pushing those outcomes or those non-desirable outcomes in the right direction and then see the outcome of that. And so it's quite a fun process to go through because you might push hard on a single EBV or trait, and then you'll see possibly an antagonistic trait then go down. And it's a real balancing act to make sure that you're finding that sweet spot with all the different requirements that you have for your breeding program. But at the end of the day, MateSel produces a crossing list that accommodates all those decisions and targets that you've placed upon it. So how does MateSel work? It is a highly complex problem, taking in all those requirements and putting them through a single decision framework. It does use a mixture of stochastic and deterministic algorithms that compete with each other. So that second point there, where we say we borrow from nature and we breed the best solution, really relates to the stochastic algorithm that we have in there. That also competes with the other deterministic algorithm. So it's not just one algorithm that we put all our eggs in one basket. We have lots of different algorithms that compete with each other to make sure that we're hitting an optimal crossing or mating solution for your breeding targets. You can integrate MateSel with other software, essentially MateSel being a solver for your crossing or mating plans alongside other systems. So you can see here, of course, ASReml is at the top of this list as well, but you can absolutely take the information as in pedigree, GRM, all the EBVs and genetic markers, and we'll feed those through to MateSel, which can then you can set your targets and where you want to take your breeding program. And once you've got that balance, you can send back the crossing plans, trait responses, and the frequencies for allele and genotypes back into those systems as well. So MateSel is at the heart of it an OCS software with a lot more on top of it. And it's good to point out there are other OCS options out there as we've got listed here. In terms of what MateSel offers in addition to those is the interactive graphical user interface, which we'll see in a second. multiple trait and EBV target settings, along with different ways to fine-tune the histograms for EBVs, genomic data support, automatic imputation for missing GRM elements for those who want to use the GRM facility. We've got some advanced features which we won't mention too much here. We'll leave those for, we've got a good website you can have a look at for those. And, we're continually investing and offering support for MateSel as well. Okay, so we're going to jump into the demo now. We're going to have a look at balancing genetic gain and diversity. We'll have a look at managing markers. We're going to also create some crossing groups. So we're going to group parents into geographical regions and see how we can control those. So I think in the example we've got today, we're going to have, I think, USA, Canada, and Europe, and see how we can control the dissemination of pollen across those different regions, and while managing diversity across the whole cohort globally. And then we'll look at the multiple end users feature, which really is for those who have multiple product markets and they want to manage diversity across all of those. So for example, you might have a nucleus, and you might have a customer that is for the pulp industry, and the other one might be for the furniture industry. So the furniture industry needs nice, long, straight trunks, and the pulp industry can take really anything and grind it up and is not really concerned with that. So we'll have a look at that to finish up. Okay, so let me just jump into MateSel. Well, before I go ahead into the demo, I'm just going to pause for any questions. Owen, Brian?
I don't see any questions yet.
Okay.
No, and
Right,
I
yeah.
think you're tracking well on time, Sandy, so you can be thorough.
Yep, sounds good. So this is the landing page for MateSel after logging in. So we've got a demonstration file here, which is called MateSel bisects demo. Now I'm going to quickly bring up what this file looks like so that you can get a bit of context for what we're feeding into MateSel. So we do accept CSV format into MateSel, but we also support a JSON format, and that's really more of a technical systems integration format that we work with software developers so that you can integrate with your other databases directly into MateSel. But for simplicity, we're just going to use the CSV format here. So I'm going to highlight the first three columns, which represent the pedigree information. So you can see we've got a candidate ID, which you could have even called genotype ID to give it more context for the plant breeding industry. And then we've got the male, the mum and the dad essentially here. If you were using the GRM, you would only have to supply this for the candidates. But what we have here is, I'm going to quickly mention this column, max use male and max use female. These essentially tell MateSel which individuals or which candidates are actually candidates. So if there's a 0, that means this particular individual is not a candidate in the data file, and they are simply there for pedigree reasons so we can work out the NRM. So you can see here that Chanel and Noah are absolutely, they're candidates for the next crossing list, okay? But if we scroll all the way down, you'll see that we've got a lot of zeros here. So these particular individuals are simply there to represent the pedigree. If we were going to use the GRM, we wouldn't need to include these individuals. We'd only have the candidates that are actually candidates. Okay.
However, it is useful to have them there because we do print out some diagnostics about the relationship between the GRM and the NRM. And also you can, for example, use the GRM for managing genomic diversity, but use the NRM for inbreeding or vice versa. So within the application, you can chop and change the use of the NRM and GRM. So it can be useful to include that pedigree information as well if you have a GRM being read in.
Yeah, and that's actually really a good point. So if you do include both GRM and pedigree, we actually do a correlation between the two so that you can do a very quick fact check to make sure that they're very closely correlated, and if there are any potential input issues. So definitely worth doing that.
And I think you don't cover it later, so I'll mention it quickly that we do automatic imputation of missing GRM elements. We do that on the fly. You don't have to ask for it to happen. And that's used quite a bit in pigs and poultry, for example. If the DNA tests haven't come in time and decisions have to be made, it doesn't stop a cohort of animals being included because the GRM elements are imputed from pedigree as closely as possible.
Great.
Yeah,
So we're
from
going to have a
pedigree
look
and
at these
other
particular.
GRM elements, of course.
We'll have a look at these fields a bit further down the track. These are these mating groups that we're going to have a look at in the third part of the demo. So you can see here these individuals have been allocated, USA, Canada, or Europe. And because plants can be either male or female, they can actually be in separate groups as well. So these are the performance metrics for each of the candidates. So you can see here we've got a selection index, and then we've also got 4 traits. So growth rate, yield, heat tolerance, and height. So this selection index here really is a weighting of all of these particular EBVs, which would typically come, from AS Remel, for example. We then
But of
have
course, it
our
doesn't have to be that. It could be independently calculated for some reason. And we should point out that this is actually a small simulated data set, and it may be a bit naive in parts, but it's illustrative.
Yeah. And then we've got a couple of genomic markers here. So we've got the height QTL and we've got rust resistance QTL as well. So essentially, that means there's been DNA tests on these particular individuals and we're representing those particular genotypes. Okay, so now you can see what's going into MateSel. Let's jump in and get a crossing list running. So when you first go into MateSel, the first question you're asked is whether you're going to be grouping or not. So we're going to just start with the no grouping scenario to start with, and then we'll have a look at grouping later. So any male can be crossed with any female. How many crossings are required? We just put a number of crossings simply there. And we then jump into the crossing policy. So really, this is an option that is not really used too often. The default option for a female plant can use pollen from a different male plant is typically always used, but we do have other options to put a bit more control on who can be crossed with who. We also have the option to avoid duplicate crossings, reciprocal crossings, and self-crossings. So they're also options if you need to put those in place.
And of course, that you can use that tool to totally avoid those types of crossings, or to put pressure against them, or even for them if you want to. It's not all or none.
Yeah. Okay, so these are the male and female candidates that we've pulled in through the CSV. So you can see that they've got their overall index from top to bottom sorted here. And now we have the maximum, minimum, and must use matings. So essentially for the male usage, the maximum matings, I've also seen this called frequency before. But essentially, it's the maximum matings that can be used for this particular individual. So this is typically set by how much pollen you've got available. And the max matings for female is typically how many flowers you've got available for them as well. So what we can do here is we can also set a minimum mating. So essentially, if I put 20 here, it means that if Salvador is selected in the crossing list, they must be used at least 20 times. However, if I put this must use option on, it means it tells MateSel that this particular individual or genotype absolutely must be used at least 20 times, at least for 20 crossings in the crossing plan. Okay, so you can control the usage of male and female across your candidates using these controls here. And that's all we need to kick off a MateSel analysis. So we'll get started with that. Okay, so we now have the MateSel analysis running, and the first thing we're going to look at is the frontier. So this is the frontier graph here, and it really is a summary of the information here. So You can see here across the crossing plan that we've currently got, we've got an average progeny index of 195 and an average progeny inbreeding of about 2.5%. Now, if we have a look at the crossing plan, we can see here the crossing list that MateSel has come up with. So just remember how MateSel works is it is constantly looking for better and better crossing solutions based on your targets. So you'll see these histograms moving around, and you'll sometimes see the frontier moving as it is developing better and better crossing plans. Now, I haven't applied any targets yet, so really all it's doing at the moment is trying to push for highest gain based on my chosen balance here, which I'll explain now. So MateSel has
And you could
calculated.
probably make the picture a little, one click bigger, Sandy. I think you've got room for that.
Sure, yeah. So we've got this thing called the frontier, which MateSel has calculated here, and it is essentially just figured out how far you can go in each direction based on the two components of average progeny index and parental co-ancestry, which is essentially what we call the long-term inbreeding. And so when we've got this particular control, we can decide where we want to be on this particular frontier. So to explain that a bit more, I'm going to go for maximum gain for this crossing list. And I'm going to have a look at our male use summary and show latest matings. So what this has done is we have essentially told MateSel that we really don't care about diversity. We just want to put the highest genetic merit through in the next crossing plan. And so what it's done is it's essentially selected the two highest indexing well, it's basically selected the highest indexing males and females to go into the next crossing plan. And look, that's great, but in terms of average parental co-ancestry, it's quite high. So if we have a look at the other extreme and go for maximum diversity, essentially what we're doing is as we go further and further towards diversity, MateSel is going to be pulling in more and more candidates to make sure that we can fill that diversity need. But to go to the extreme, if we go to full diversity and show the latest mating solutions, you can see now that the mating list is pulling in a lot more males and females to achieve that diversity. But of course, we want a balance of that diversity. So, we look along this frontier line to figure out where we want to be. Where you want to aim for
And
on this
before you
particular.
update that, Sandy, just quickly that you'll see that even though the bars on the right are for going for full diversity, you've got some individuals being used a lot more than other individuals, and that's because they're less related to the rest of the candidate population, and they've got more to contribute in terms of diversity. So it's not just equal contributions; it's optimal contributions.
Yeah, absolutely. And we have the male use detail to help with that as well. So you can see here we've got the co-ancestry for the candidates and then the co-ancestry for the selected candidates. And you can see they're quite well balanced across all males there. Okay. We also actually have a lot of online videos as well, which explains the frontier and all the other features as well. I should have mentioned that as well. So within the application and within our documentation website, which is mateseldocs.com, you can actually view videos on how all of these controls work and get a bit of an explanation on where you should aim on this particular frontier as well. Okay, so once you have decided and set a policy for your breeding program on where you want to be in terms of balancing your index with parental co-ancestry, you then turn attention to the traits. Okay, so these trait histograms represent the expected progeny outcomes for each EBV. And essentially, it comes back to these crossings here. So these are the crossings that MateSel is suggesting to make. And if you were to make these particular crossings, then these are the trait outcomes you would have in the progeny. So you can see here, we've got this little black tick here, and then a red tick here. So the black tick represents the candidate average, And the red tick represents the progeny average. So if you were to take this crossing list, this is what the expected outcome on average will be, and then simply the response of that particular trait. So with that in mind, you can see that we've had a positive response for everything except for heat tolerance. So let's say that we wanted to maintain our level of heat tolerance with the candidate average. So you can see we've got a negative response there. We can actually manipulate the crossing list to say that, okay, I want MateSel to at least hit that target there. Okay, so what you can see is MateSel has now come up with a new mating list. Well, in the background, it is actually coming up with millions and millions of new mating lists, and only the best are being promoted. But you can see there that we've now been able to modify the outcomes, so we are now maintaining that candidate average in the crossing list. Okay, now if you were to be very aggressive and say, actually, I want really, really high response on heat tolerance, have a look at what we've done to the index on the left-hand side. So yes, we can achieve this particular heat tolerance EBV target, but we've made a compromise on index with probably 4 index points, which is fairly significant, okay? So it's really important when you set these targets to always have a look at what impact you've had on your index and co-ancestry, and maybe start backing things off until you've got a happy balance and that compromise in index is acceptable, okay? Other things we can do is if this is the height of the plant, we might feel that, we want to have the height. We don't have any dwarf plants, so we might set a minimum value for this particular EBV and apply that. And we'll just wait for MateSel to finish evolving. So we've now got that, but we've now got quite a few heights. It's got a lot of variation still within this histogram. So we might want to decrease the variation to make sure the height's more uniform. So instead, what we can do is we can decrease the variation, update that target, and we'll just see MateSel evolve to a solution where it's essentially topping and tailing the height for the mating lists or the crossing lists, and pulling that into a more uniform, reducing the variation there. So you can see we can have multiple trait targets, and that's quite easy to do. And if we remove a target, you can then see all of that variation start coming back. Okay. So after you've tweaked your traits, so we've now figured out our balance here. We've now got the traits to where we want them to be. We then focus our attention on inbreeding. So this essentially is just the inbreeding coefficient across the whole of the crossing plan. So I can also see the impact of this by having a look at the crossing list. You can see it's still evolving a little bit there, but you can see we've got most of our matings at around at most 3%. So if we have a look at the latest mating plan and sort by progeny inbreeding, there are those 3% inbreeding items that we can see on this histogram. Now, if we were to say that, we actually need to set a limit on let's say 2.91, and we'll update the target. The MateSel will now rearrange the mate allocation and also the selected parents if needed to achieve that. So it can take some time to get to that target. But what you can do is if you find that there are multiple competing priorities and you need to push one target higher than the other, then we've got these emphasis options that allow you to push a target higher than another target. So in this example, we're not quite getting to where we need to, and I'm going to hit that on a stronger emphasis, and it's telling the algorithm to prioritize that as a target. So you can see now we've actually hit that target. Small compromise in our overall index. Again, you need to review that after every target. But if we go back and look at the latest solution, you can see now that the progeny inbreeding is not above 2.91. I'm going to take that target off, and just out of interest, something that you can also do is you can also increase inbreeding if you want. So I'm going to just do that as a moderate emphasis for now. Actually, let's just push that up. So in reality, this is not something you would do, but it sort of shows you what you can do. And if you were to look at the crossing list now, you can see we've got a lot of selfings happening, and that's where that inbreeding, that high inbreeding is coming from.
Yeah, and
You
you
can
could
see
see
that.
full set mating. They probably disappeared now, but you could see the full set matings and half set matings were there as well. It's
Yeah,
now all
yeah.
selfing.
So there are actually some applications out there for some MateSel users to be able to do that, but typically you're going to be wanting to reduce your inbreeding.
But it does show, of course, that you can control. This is a rather crude way of controlling proportion of selfings. You can do that more directly, but we've got other better ways of doing it where you've got different parts of your breeding program where you're doing outcrossing and selfing, and we'll talk about that later.
Yeah. Okay, so we're going to move on to managing markers. And so here we've got two QTLs or markers for height and rust resistance. Now, these can be gene edits. And essentially, we're going to be trying to do two things with rust resistance. The first one is if we have a look at the 2-2. So let me explain a bit further. Sorry. These histograms represent the genotypes. These bottom four represent the genotypes. So you can see we've got the 1-1 homozygotes, we've got the 1-2 and 2-1 heterozygotes, and the 2-2 homozygotes. The first chart here represents the allele frequency of the favorable allele. So for this example, we're going to assume that the 2-2 genotype for rust resistance is the undesirable. We do not want any crossings which result in a 2-2. So what you can do is you can jump into that histogram and put a decrease progeny value, which is going to push the frequency of those of the 2-2 genotype down. So I'm going to apply
Just
that
to
now.
clarify what you're seeing there, the small blip in the middle there with the 0.5 means that number of plants of crossings, say 10 or 15 crossings, have a 0.5 chance of being 2-2 genotype. So that what we're looking at is the probability in the progeny of being of that genotype. And of
Yeah.
course, that's all gone down to zero quite nicely.
Yeah, absolutely. So that is a simple way of pushing the 2-2 genotype out for the progeny. Now, another thing that you can do is if you want to increase the frequency of the favorable allele, you pretty much just do the opposite. So you can increase the average progeny value for that one as well. Now, like I put an extreme weighting on this particular one here, so it's not going to compete very well at a moderate weighting. So I'm just going to put that up. And you can see MateSel is almost there. It's there now. But again, this is probably an unrealistic scenario in your breeding program to push so hard in one generation. You'd most likely just try and ease that off over generations to make sure you're pushing the favorable allele, a little bit for each generation, so that the compromise and overall index isn't going to be so extreme. So
And a
again,
typical
this
thing
is
to do is to set a target for that red dot year by year or generation by generation. So you've got a plan for the change in allele frequency in key edits and so forth over time. And you do everything else you can to have a good breeding program given the constraint that you want that change. And of course, you can see the compromise you make in achieving that sort of level of change, and you can change your plans. But it doesn't have to be just how hard you push. You can actually target a specific outcome for all of these things. And that's already been shown for one of the traits.
Yeah, absolutely. So I'm going to just remove these targets. So that wraps up the initial demo. We're going to jump into looking at grouping. So I'm just
Do
going
you
to
have time
remove.
for a couple? There's a couple of questions that have popped in. Is that our
Yeah, yeah,
time to?
absolutely.
Yeah.
Yeah,
Okay.
I've been looking at those, so I could probably answer because I've had a chance to look at them while Sandy's been talking. So
Yep.
is MateSel a closed system or would our data go anywhere out in the world? No, MateSel is an in-house system, so your data doesn't go out in the world. You've got full control over all that. The next one is, could you please elaborate more on the index calculation? Well, the index calculation is an input to MateSel, so that's really something which you do before getting to MateSel using other tools, and there are good ones around. And it is just your overall a impression of how useful each individual is, like a dollar EBV is another way of thinking of it. So we have that as input. The next one is how is it handling multiple phenotypes if only one GEBV is being provided? I think what we're talking about there is probably using the genomic EBVs based on the GRMs. That again is input. The EBVs that come into MateSel can be pedigree based, genomic based. You can use separate GRMs for separate GEBVs, taking account of QTL. All of that is input to MateSel. We handle it once we get it. And is the algorithm under MateSel working different crop types, inbred, hybrid, clonal crops? We'll address that in just a moment. That's Auburn. Sorry, Auburn, not to answer at this stage. And lastly, Aristide, is there a range for the index? And the answer is no. That's totally up to you. It is, again, your overall impression of the merit, and we don't put any constraints on the range in the index. Okay, I think that's all that's on the chat at the moment there, Owen.
Yes, there has one or two pop up in the Q&A, but maybe how much time do you need? Do you want to come to those at the end if we have time?
I mean, we can address them now. That'd be good. Because we're going pretty good for time, I think.
Okay, great. So from Martin, we've got, does the algorithm to stimulate crosses need a genetic map of the genetic data? Question for soybean.
Can you say that one more time, please, Owen?
Yeah, sure. So does the algorithm to simulate crosses need a genetic map of the genetic data? Question for soybean.
No. Again, it's input. That sort of a material is calculated beforehand going into
Okay,
MateSel.
thanks.
Yeah.
Can QTL major genes be stacked? Haplotype selection slash planning of crosses.
Yes, good question. We do have a means of managing uh, non-additive components, and so we've been working with Tobias Nijhoff at Wageningen University for look-ahead mating systems and managing Mendelian segregation variants. We are open to anyone who's got algorithms that they want to have for haplotype stacking as to how to give favor to rare alleles which are only represented in certain candidates. We have a route plan to develop that within MateSel, but there are multiple ways of doing that. We want to be able to have a plugin with any other external software for calculating how to do that. In the end, I think we'll offer a range of options for that. That is where the value of a mating depends on what other matings are made because of rare alleles and haplotypes. So the answer is a bit mixed on that one, and we'll be in a position to do something quite soon on that.
Fantastic. Is there any computational requirement?
Yes, a computer.
Yeah, look, I mean,
Uh,
the bigger your RAM and more importantly your CPU, the faster that the algorithm will run. We definitely recommend an i7 at least, but it all depends on how many candidates you're feeding through the system and how many crossings you're requesting. It's a function of that.
And then can we use LD-based haplotypes with the MateSel algorithm?
Yes, it depends what you want to do with them, of course. Many things, if you're using them to get more accurate estimates of breeding value, obviously yes. If you're using them to predict Mendelian segregation variants, or if you're going to predict embryo mortality and therefore fertility in plants or animals, yes, we have a very special system under a USDA project we developed for handling multiple haplotypes and markers collectively. So there could be hundreds of those things, and we have a simple way of managing them, and we've done that already for recessive lethals, but we have a route to doing that for collections of gene edits. We're not The industries are not there yet with such large numbers of gene edits to our knowledge, but we are ready to handle that. I don't know how well that answers the question.
Yeah, that's great. Fantastic. Thanks, guys.
Okay. Thank you. I'll probably just quickly add as well, you don't have to use the graphical user interface with MateSel. We do offer what we call the batch version. And a lot of people use that in simulations and other pipelines. So essentially they just automatically, pull information that they need from MateSel, and that's in an automated scripts. And then MateSel runs as a background or batch job, and then delivers the output. So you don't have to go through the graphical user interface is the point there.
Okay, just
Okay,
a quick
so
point. We may be running a little bit later in time now, so we'll probably have to keep cycling.
yeah, we'll be pretty quick on this one. So the grouping feature is very quick to demonstrate. So let's quickly jump into that. So you saw in the data file, we had a group for males and females, so USA, Canada, and Europe. So these have been pulled through into the interface, and you can see in terms of the male groups, we've got two with 74 individuals each. And for the female groups, we've got Canada, Europe, and the USA. So essentially, you can set up the same max matings, minimum matings that we saw with the no grouping scenario, but they'll apply to all of the individuals within the group. You can actually set them individually as within this group feature as well, but we'll not do that in this demonstration. So once you have your groups defined, you now get this matrices of essentially what we call the permission matrix. So in this example, what we're trying to achieve is no transatlantic migration of pollen. Various reasons for that. So we've got our pollen in Europe and the USA, and we need to disseminate that across different regions appropriately. And you can see here from the Europe, we're only going to be disseminating out to Europe. From our US operations, we'll be pushing out to Canada and the USA. And we're just asking for 30 crossings in each of these particular combinations. So we'll just hit next and run the
And we could
MateSel
run
analysis.
separate breeding programs for these, but the reason that we don't is we want global management of diversity for this single line, because it could be that in a year or two's time, there's going to be a quarantine program or whatever. So this may be a bit of a naive example, but I think you can probably have situations in your head where you can see how grouping could be used.
Yeah, I think these could be plots potentially within different regions. It's essentially, this is the only difference. You can now see that MateSel's adhered to those particular grouping constraints. So if we had a look at USA, you can see that the pollen's going out to a mixture of Canada and USA. However, when we get to Europe, we're only going out to Europe. So you also have a number of dashboards. You can see here a breakdown of each of the plants and where they're being used. And you can also have a look at the group summary, just to see that, for this particular male group, the average index selection before and after selection. So you can just make sure that, we're heading in the right direction, generally with your selection index. Okay, so I'm going to stop the run there, and we're going to jump into the mixed mating groups. Now, I'm just going to bring up the PowerPoint while I just get this ready on my other screen. Just one
Yeah,
second.
well, mixed mating groups actually, I think, it was a slip of the tongue, can be used for managing cohorts of individuals, and we're not going to talk about that one today. We're talking about multiple end uses. So this is where you've got a number of different end uses, typically, but not exclusively, it's for different traits. So it could be that you've got high versus low demand in different regions or client groups. for particular traits like drought resistance, oil content. And the example on the right of the trees is where you might have pulp and furniture as two distinct or relatively distinct markets. And what you could normally do is just in your nucleus, you go and get good pulp individuals and good furniture individuals to send off seed stock to those markets. We're operating a generation before that because what we're doing here is making matings which are going to give you a better distribution of your end use needs, in this case, pulp and furniture, as well as the nucleus being an end use as well, and a very important one, which you don't want to compromise too much in what happens in the nucleus as far as your dissemination program is concerned. So it could also be a specific disease important in some regions or others. Obviously, G by E, where you're targeting different environments coming out of the same breeding program. And to some extent, also different breeding program roles, so that you want so many matings to be selfings as opposed to outcrossings of some kind. And that's part of the overall design of your breeding program. So you can make MateSel actually work with some strategic design you have for the breeding program. It's not just traits. You can essentially identify groups for intensive phenotyping or genotyping. And the example there is the genomic reference population. And for that one, what you're really looking for is to have individuals that are quite diverse, not necessarily of high merit, but quite diverse because you're going to use them for genomic calibration. So you want to have low co-ancestry within that reference population, and yet simultaneously you want to have high co-ancestry between the reference population and the population that you're breeding in. And so with MateSel, you can declare different populations and manage the co-ancestry within and between. So that's the degrees of relationship within and between populations in order to achieve that. So that's a key example. But the next and last one there is that we can, if we were wanting to spin off experimental programs, so you could say 10 or 20 crossings for high oil content and 10 or 20 crossings for low oil content, you want to make those crossings for an upcoming experiment. We can manage it so that the co-ancestry between those two experimental groups is high, so you've got more power in your experiment. And then, of course, you need a GenStat or so in order to take account of the fact you haven't made random allocation to your experimental treatments. But this gives you power to generate the trait profiles you need for experiments, as well as increasing power by balancing essentially stratified random across either a numerator relationship or GRM relationships. So what we'll do now is go ahead and give you a demonstration of how that works in a simple scenario.
Yep, absolutely. So, okay, so we've had to just flick over to a different version because the multiple end users feature, although it's been around for a couple of years, we've just released a new graphical user interface, and we haven't released the multiple end users feature. So we're running on the on a beta version at the moment for multiple end users. But let's just jump into it. So we've already gone through this screen here. So let's jump over to multiple end users. And what we've got is we've got a scenario where we've set up a nucleus and we've got two essentially end users. So we've got the drought risk and we've got the temperate region that we want to satisfy. So within the nucleus, we really want to make sure the best genetics are going back into the nucleus. And we also want to make sure that co-ancestry is being considered going back into the nucleus as well. Whereas with the drought risk, we need to put an emphasis on the heat tolerance EBV. So what we're really saying there is we're really focusing on that particular EBV being quite high for this particular region. And for the temperate region, we are looking at growth rate. That's just a random, it's probably a bit naive as an example, but it's just a demonstration to say that we can push different traits and markers for different end users, essentially.
And in reality, you'd probably push and pull all traits for all regions in different amounts, but this is for simple illustration.
Yeah.
It doesn't have to be just one thing for one end use, of course.
Yeah. So remember that was going to happen. So I think that should do it. Oh.
Number of matings.
Just 1 second. That's always the risk when you're doing these things. Okay,
I've got it here, Sandy.
I'm just gonna just quickly stop sharing. I will be back in just 1 second.
Okay, anyone got any questions on what we're doing with multiple end uses?
I can see the
Well,
problem.
don't. Oh, right.
But yeah, if there's any questions, now's a good time.
So what you're going to see is the outcomes we have for all those different components. And in particular, we want to manage diversity in the nucleus, but the drought risk and temperate, we're seeing these as dissemination end uses so that we're wanting to send that material off for dissemination to create varieties and so forth that are not going to be coming back. The results of these matings are not going to be coming back into the nucleus. Now, that's not necessarily the case. If there's a probability that some of that material can come back, we can put some emphasis on diversity for those other end uses. There we go.
Yeah, sorry about that. Like I said, it's a beta version, and I knew there was one issue with it, but I wasn't expecting that one. So that's all good. Something to fix up a bit later today. So let's just recap what we're doing here. So we've got the nucleus, drought risk, and temperate. If you see a green number here, that's essentially the average for that end use. And we'll actually just show all traits so that we can get an idea of where we are. If you see a blue pill, essentially, it means that we've got pressure on that particular trait. So you can see here on the index for the nucleus, we've got a push on push up and heat tolerance for drought and growth rate for temperate. So what we expect to see here is that the index for the nucleus is going to be higher than drought risk and temperate end uses. And you can see we're running at an average progeny average of 191 versus 177, 181. So that's great. So that end use is doing what it's supposed to. We're also seeing that the heat tolerance EBV is well above the averages for the other two end uses. And we can see growth rate is above the other two end uses. So a very simple example on taking different end uses and pushing them in different directions from the one population.
And
You can
the
see
low
also
co-ancestry emphasis has worked nicely too,
yeah, so we put low co-ancestry emphasis on that one, so you can see that's quite a bit lower as well.
compared to the two to the right.
Yeah.
Yeah.
Okay, so you might have a look at this and go, well, the index is bigger, but I need it to be bigger than that for the nucleus. So we might put a higher emphasis on that and update the end use. And so that was 192. So you can see it's slowly creeping up. And we can put a higher emphasis on that if we need to push it further. But they're the sort of controls that you can have live on screen as well, just to start looking at the averages and start pushing them in higher with higher emphasis or lower emphasis, and also adding other constraints like markers. So if we show all the markers, you can also see the average of the allele and genotype frequencies. So you can also have a visibility on how they're tracking within each end use as well. Now, this frontier is a bit different because this is the frontier across all end uses. And that happens for traits, markers, and inbreeding as well. So if we were to come in and set a progeny inbreeding limit here and apply that, that's actually working across all three end uses. Okay, so we now know that within all end uses, we won't have any crossings which result in above 4.8% inbreeding coefficient. We'll just head back there.
And
Okay,
it won't be as pretty as this, but you can hop online and look at our video. We've got a set of videos on managing multiple end uses, and there's more power than what you see here, and you'll see that in those videos. But this is much easier to use once you get it going.
Yeah, absolutely. So I think that really gives us a very broad brush on the features of MateSel. Like we said, there's a bunch of other advanced features that we haven't looked at. So we would encourage you to head over to mateseldocs.com, or you can get there from matesel.com as well. It's just so you can do a little bit more reading if you want to understand a little bit more about each of the different features we've got. There's also links to all of our videos for genomic mate selection, mixed mating groups, and all the other things that we didn't get a chance to demonstrate today. But yeah, we definitely encourage you to have a look at that. But that really concludes the demo, Owen. So just a big thank you for everyone for sticking around if you're still there. And yeah, we're always open to hearing feedback and questions. So yeah, happy to field any questions after the webinar as well.
Fantastic. Thank you very much, Sandy and Brian. That was brilliant. It's always great to see the tool in action. I can imagine a lot of people are licking their lips at the opportunities now, thinking about their breeding programs. Um, we've had a couple of extra questions pop in. If you've got time for one or two before we finish.
Sure.
Yes.
Sure. Okay. So we've got. Is there a Linux version for MateSel on for HPC?
There is, yeah. So we do have a Linux version for the batch version, so for simulations or running it through pipelines. The graphical user interface version is currently only available on Windows, but that might be changing in the near future.
That's it. Someone's also asked, can we do simulations using marker data without linkage map? Let's say we genotype the whole population using genotype biosynthesis.
Genotype.
Without a linkage map. Well, certainly you can do simulations with a linkage map if you've got unlinked markers, if they're taken to be unlinked.
Sorry, I might
Yes,
have mispronounced without a linkage map.
without a linkage map.
Sorry.
Yes,
Yeah.
any weaknesses from not having a linkage map would be reflected in anything you do, including MateSel, of course, if you don't have the information. But yes, there's quite a large number of groups and in-house companies who do simulations and looking at what happens for everything, including markers. I don't know if I'm really answering the question. I'm not exactly sure how the lack of a linkage map is impacting here. Maybe that can be refined, that question.
I mean, we do have quite a lot of documentation on mateseldocs.com, which is related to how you can use genetic markers within MateSel. So it'll take you through the options in terms of using genotype probabilities, where you've only DNA tested a subset of the population, or if you have everything genotyped, then there's that option as well. So, and if that doesn't answer those questions there, we can definitely jump on an email and get that answered for you as well.
Yeah.
I guess we'll have time for one last question from Paul Coyne. Is this model primarily applicable to single crosses as opposed to pooled spawns, i.e. aquaculture 30 size by 30 dams?
Yeah, well, what we haven't shown you is our mixed mating groups. In fact, we could almost shove something up, but I don't think we've got time. We've got a very rich feature where we can handle mass spawning in fish, multiple joining in cattle, open pollination in plants. But importantly, we can handle grouping and cohorting of individuals using the same approach through a project so that, for instance, if you've got multi-stage selection and you're wanting to identify individuals to send off to test plots for higher levels of phenotyping, that is actually coming into like pooled spawns. And of course, I'm focusing on plants probably too much here. Sorry, Paul. But that has been used quite in a couple of the pig breeding companies now to manage the whole operation, migration of animals between different types of breeding units. And that can be done in plants. When I presented that in a seminar, a plant breeder up the back said that looks quite good for us. So we haven't heard of that being used in plants yet, but that's a whole other arm of MateSel, which we hadn't got a chance to talk about yet.
Fabulous. Thank you very much. I'm aware there's some questions that haven't been answered. Brian and Sandy have kindly agreed to write up some answers to those so we can send that out along with the recording once that's all been uploaded. So we'll get those answers out to you. If you would like any more information, please feel free to reach out to any of us. You can reach VSNI through the info at VSNI or through our contact form, or feel free to reach out to me, Sandy, or Brian.
Absolutely. Thanks very much, everyone. Really appreciate it.
Yeah, thanks. Great opportunity. Good working with VSNI.
Yeah. Thank you very much, Sandy and Brian. That was a fantastic presentation. Cheers. Thank you, everyone.
Cheers.
Bye.